Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Delhi University MSc Entrance Examination for the subject Plant Molecular Biology and Biotechnology conducted in the year 2021. Coming to the first question. Virus-free plants can be obtained by meristem culture. Answer is option 1. Everybody knows that answer. Botanical name of pigeon pea is Cajanus cajan. Answer is option 2. The famous plant breeder Norman E. Borlaug received Nobel Prize for Peace. Answer is option 1. Spirulina is a blue-green algae or a cyanobacteria. PCR was invented by Kary Mullis. Then Murashgi and Skoog are famous for plant tissue culture medium. The experiment showing that the tip of the coleoptile senses light was performed by. So it was performed by Darwin and C. Darwin and F. Darwin. The chemical derived from Taxus brevifolia is used for. It is used in cancer therapy and it is called as paclitaxel. It is derived from the bark of the Pacific yew tree and is used in the treatment of breast, lung and ovarian cancers as well as Kaposi sarcoma. So answer is option 2, that is cancer therapy. Agent Orange herbicide used by the military forces during the Vietnam War contained which of the following plant hormones? So it contained auxin because it had the components 2,4-D and 2,4,5-D, both are auxins. Then gram staining in bacteria primarily stains the cell walls. An example of a scorable marker in plant. So scorable marker are reporter genes that generate generate a product that can be detected using simple quantitative assays, whereas selectable markers are detected through the expression of the resistance for a toxin or a resistance for an antibiotic. These are selectable markers. So Gus is the scorable marker here. Then isozyme analysis can be performed with which of the following polyacrylamide gel types? So isoenzymes, they are a group of enzymes that catalyze the same reaction but have different forms, catalytic efficiencies, and they are usually distinguished by their electrophoretic mobility. So by choosing native or non-denaturing page system, you can separate protein based on their size, shape and net charge of their native structure while preserving their function and activity. So non-denaturing gel. Answer is option 3. Then 1 gram of which of the following molecule can be produced maximum amount of ATP upon complete oxidation? Naturally it is triglycerides. Answer is option 4. S-adenosyl methionine is the S-adenosyl methionine is the precursor for the biosynthesis of putrescine. Putrescine is a polyamine. Answer is option 4. Then which of the following is not a reducing sugar? Sucrose is not a reducing sugar. Then next one. Why are polysaccharides better suited to store energy in a cell as compared to monosaccharide? So if glucose is stored inside the cell without polymerization, then the internal environment of the cell will be hypertonic, causing osmosis of water into the cell. Ultimately, the cell swells and it gets led to the lysis of the cell as a result of osmotic pressure. Therefore, it is advantageous to store glucose in the form of starch or glycogen. So the answer here will be monosaccharides would create excessive osmotic pressure. Answer is option 2. Then next one. Which of the following is an amino derivative of a hexose sugar? So it is sialic acid. Sialic acids are class of keto acid, alpha keto acid sugars. Answer is option 4. The main site of synthesis of cross-linking glycans and pectins of plant cell wall is. So it is Golgi apparatus. Answer is option 2. Then which enzyme is the prime target of the commercially important herbicide glyphosate? It is enol pyrovyl shikimate 3-phosphate synthase. Answer is option 3. The biosynthesis of aromatic amino acid in plants primarily take place in. It takes place in the plastid. Answer is option 3. Which of the following amino acid is the precursor for polyamine biosynthesis in plants? It is arginine. Answer is option 3. Fatty acid biosynthesis in plants take place in. So, fatty acid biosynthesis in plants. Yes, in plants it takes place in chloroplast. Answer is option 1. In animals it occurs in the cytosol. The next one. Quantum yield efficiency of 1 during photochemical reaction of photosynthesis indicates that. Okay. So, the quantum yield of a photochemical reaction describes the number of molecules undergoing a photochemical event per absorbed photon. That is, number of molecules undergoing the reaction of interest divided by number of photons absorbed by the photoreactive substance. So, in photosynthesis, it is micromole of carbon dioxide fixed by micromole of photon absorbed. So, a quantum yield of 1 describes a process where each photon absorbed results in a molecule of carbon dioxide being fixed or result in a photon being emitted. So, the answer here is option 2. Every absorbed photon is converted into chemical product. Answer is option 2. Then next one. Cyclic photophosphorylation produces only ATP. It is cyclic photophosphorylation, so it produces only ATP. QTL represent quantitative trait locus. Answer is option 2. Then ENCODE expands to. It expands to Encyclopedia of DNA Elements. Answer is option 1. tRNA is synthesized by. It is synthesized by RNA polymerase 3. Introns are removed at the level of RNA. Then which of the following scientist was awarded the Nobel Prize for DNA sequencing? So it was Gilbert. W. Gilbert. Gilbert was awarded Nobel Prize in 1980 in chemistry, shared with Frederick Sanger and Paul Berg. Then next one. Activation tagging involved. So activation tagging randomly inserts a tDNA fragment containing engineered 4 copies of enhancer element into a plant genome to activate transcription of flanking genes. So, activation tagging involves tagging a gene by its activation. Answer is option 2. A nonsense mutation involves creation of a stop codon. Answer 4. Mobilization of retrotransposons involved. So, retrotransposons, they move by copy and paste mechanism. The copy is made of RNA. The RNA copies are then transcribed back into DNA using reverse transcriptase and then are inserted into new locations in the genome. So it is a copy and paste mechanism. So both RNA and DNA form. Answer is option 2. Small nucleolar RNAs are involved in. So small nucleolar RNAs are non-protein coding RNAs and their main function is to guide site-specific rRNA modifications. So they are involved in rRNA processing. Answer is option 1. Nucleosome is made of. So nucleosome is the basic structural unit of a chromatin and it is made up of a coil of DNA wound around a histone core. So it is made of histones and DNA. Answer is option 2. A plant cell contains DNA in. So in plant cell it contains DNA in 3 organelles: nucleus, chloroplast and mitochondria. What is the maximum resolving power of a light microscope? It is 200 nanometers. Answer is option 4. Then, which of the following microscopy is used for live cell imaging? So, it is phase contrast microscopy that is used for live cell. Live cell imaging is phase contrast microscopy. High magnification of electron microscope is due to, it is due to electromagnetic lenses. Next one. Which of the following organelles exhibit highest succinate dehydrogenase activity? So, answer is mitochondria because succinate dehydrogenase is present in the inner mitochondrial membrane. Then, in plants, the process of photorespiration takes place in the following cellular organelles. So, it is mitochondria, chloroplast and peroxisome. And option 2. What is the primary role of CDC protein in the cell? CDC is cell division control protein. So naturally its primary role is in cell division. Answer is option 3. Then which one of these statements is true? Plant vacuoles have their own genetic machinery. No. Plant vacuoles are considered as versatile lysosome. Yes. Plant vacuoles are involved in respiration. No. Plant vacuoles are involved in biosynthesis of lipids. No. Then next one. Tonoplast is a membrane of which organelle? Of vacuole. Answer is option 2. Middle lamella is made of pectin. Pectin is chemically glucuronic and galacturonic acid. Answer is option 3. Which organelle is not involved in vesicular transport in eukaryotic cell? So it is peroxisome that is not involved in vesicular transport, whereas others are involved in vesicular transport in eukaryotic cell. X-ray crystallographic analysis of DNA was performed for the first time by Rosalind E. Franklin. Answer is option 1. The method which played a crucial role in deciphering the genetic code, it is the filter binding assay using radioactive amino acids. Answer is option 3. Then HFR strain in bacteria is one in which the F plasmid is integrated in the chromosome. Answer is option 4. Which of the following statement is true for bacterial promoters? -10 region is located 10 base pair downstream. No, it is upstream of the transcription start site. Then -10 region shows more conservation than -35 region. That is correct. Then next one. When E. coli is grown in a mixture of glucose and lactose, it catabolizes. So it will first utilize glucose. E. coli has more preference for glucose than lactose. So it will first utilize glucose until it is exhausted, then lactose. So the answer here is option 2. That is first glucose till it is exhausted, then lactose. Then next one. Transfer DNA from a Ti plasmid is maintained in a transgenic plant by integrated DNA in chromosome. Answer is option. Then next one. Lederberg's and Tatum's experiment demonstrating the exchange of genetic material in bacteria used. So they used auxotrophic mutants. Answer is option 1. Golden rice is the name of a genetically engineered rice containing genes derived from. So they are derived from daffodil. Answer is option 4. Then phytoalexins are plant secondary metabolites which show diurnal cycle. No. Compounds produced by plant in response to pathogen attack. Yes, that is phytoalexin. Next one. Which of the following has iron as part of its structure? So it is cytochrome. Answer is option 3. Then telomerase complex involves the following. So it has got the reverse transcription and template RNA. Answer is option 3. In bacteriophage lambda, the repressor protein acts to promote. It acts to promote lysogeny. Answer is option 1. Ethyl methane sulfonate causes mutation by alkylation of bases of DNA. Answer is option 1. Then next one. Which of the following antibiotic results in translation termination? So alpha-amanitin, it has got different effects on various RNA polymerases. Then ampicillin, it inhibits cell wall synthesis. Puromycin is a premature termination of translation. And streptomycin, it irreversibly binds to the 16S rRNA and S12 protein within the bacterial 30S ribosomal subunit, and as a result there will be inhibition of initiation of protein synthesis. So what they have asked, which of the following antibiotics results in translation termination? So it is puromycin. It results in premature termination of translation. Answer is option 3. The ploidy level of endosperm is 3N. Option 3. Scutellum in the cereal grain represents cotyledon. 3. The alluron layer is a source of that question was dropped. Okay, this question was dropped by the university. The principal nitrogen fixing algae in paddy field is Oscillatoria. Who is credited with advancing the science of paleobotany in India? So it was Birbal Sahani. Answer is option 2. Eyes on potato tubers represent. They represent nodes with buds. Root cap is not found in hydrophytes. Hydrophytes, they don't have root cap. In certain parts of India, muscular dystrophy like symptoms are commonly found in people who consume seeds of Lathyrus sativus. Answer is option 2. Chilgosa is an edible product obtained from Pinus gerardiana. Then Casparian strips are found in. Casparian strips, they are found in the endodermis. The breadwheat Triticum aestium that is commonly used all over the world is a hexaploid. All these are just direct questions. You can easily answer them. The phenomenon of reversion of mature cells to the meristematic state leading to the formation of callus is known as, it is called as dedifferentiation. To obtain intergeneric hybrids, which of the following is practiced? So it is embryo culture. Then signaling during pollen tube growth and guidance in pistil involve Ca2 plus ions. Then which of the following statement is true for a double-stranded DNA with 40% guanine? So 40 guanine means cytosine is also 40. A plus T will be 40 and 40 will be, wait 1 minute, G plus C will be 40 plus 40, so it will be 80. 100 minus 80 will be 20. So 20% each will be A and T, or A is equal to 10% and T is equal to 10% and C is equal to 40%. So the answer here is option 1, that is adenine 10%, thymine 10%, cytosine 40%. Which of the following set of enzyme will be required for cloning a 500 base pair DNA fragment in a 3 kb vector? So it is restriction endonuclease and ligase. Answer is option 2. So what is the function of LAC-Z gene in plasmid cloning vector? So it allows identification of bacteria containing recombinant plasmid. Answer is option 2. Then in a tube, mRNA molecule and its corresponding genomic DNA fragments are mixed, denatured, and conditions were provided to promote their hybridization. Okay, so mRNA and DNA molecule, they are mixed in a tube and they are allowed to hybridize. The hybridized product showed incomplete pairing in some portions, resulting in the formation of loops. These loops are termed as. So there was no complete hybridization. There was formation of loops when mRNA was hybridized to DNA. Now these loops, this is the genomic DNA. This is the genomic DNA and this is your mRNA. Sorry, this is genomic DNA. Okay, this is genomic DNA was the one which formed loops, whereas mRNA didn't form loops. Now what do these loops represent? So they represent introns. Answer is option 1. Which of the following attributes are associated with E. coli DNA polymerase 1? So it has got only 5' 3' polymerase activity, no. Only 5' 3' polymerase, 3' 5' exonuclease, no. 5' 3' polymerase, 3' 5' exonuclease, 5' 3' exonuclease. All these activities are present in DNA polymerase 1. Then reverse transcriptase enzyme is known as RNA dependent DNA polymerase. So it requires RNA and from RNA it will create DNA. So answer is RNA dependent DNA polymerase. With regard to epigenetic regulation of eukaryotic genes, which of the following statements hold true? So epigenetics, it is the study of how cells control gene activity without changing the DNA sequence. And epigenetic changes are modifications to DNA that regulate whether genes are turned on or off. So addition of methyl group to cytosine basis of DNA, that is an epigenetic regulation of eukaryotic genes. Answer is option. Then a Drosophila, a dominant allele H reduces the number of body bristles, giving rise to hairless phenotype. So capital H, it give rise to hairless phenotype, and small h give rise to hairy phenotype. In the homozygous condition, H is lethal. Homozygous means if there is two H together, that is HH, then it is lethal. It will lead to the death of the organism. When the hairless progeny are backcrossed with parental hairy fly, what phenotypic ratio of hairless and hairy flies would you expect to find among their live progeny? So hairless progeny is H, capital H, small h, that is hairless. If it was HH, then it will be lethal for the organism and it may die off. So naturally the hairless progeny is a heterozygote. And it was backcrossed with parental hairy fly. Now hairy is homozygous condition and it is a recessive gene, so it is naturally in the homozygous condition. So it will be small h and you will get 1:1 progeny. So answer will be option 4. Then what is reverse genetics? Reverse genetic induces mutations in model organism to study their role in disease. So it is experimental approach to decipher function of a selected gene by disrupting its function. Answer is option 1 itself. Which of the following photoreceptor is principally involved in regulating seed germination? So it is phytochrome. Answer is option 2. The next one. In germinating rice seeds, gibberellic acid induces the de novo synthesis of alpha amylase in aleuron layer. Answer is option 1. Gene transfer in plant cells can be obtained by. So it can be obtained by evaporation, no. Electroporation, yes. Which of the following is used for the preparation of competent bacterial cell? It is calcium chloride that is used. Then which of the following DNA sequence is palindromic? So a palindromic sequence, it will read the same in both the strands, that is in complementary strand and the parent strand. It will read the same in the same direction, that is 5' to 3' direction. It will be the same. So coming to the first option, 5' to 3', it is TTTAA, that is 3', but in the complementary strand 5' to 3', it is not reading the same as the main parent strand. It is 4T here, whereas only 2T in the complementary strand. So that is not palindromic. We will see for the second option. Second option is AAGCTT. And if you read in the 5', 3' direction for the complementary strand, it will be AAGCTT. So option 2 is palindromic. Whereas if you see option C also, it is not palindromic. And if you see option 4 also, that is also not palindromic. So answer is option 2. The next question. Which type of restriction enzyme cleaves DNA at or close to recognition sequence? It is type 1. Sorry, type 2. Okay, it is type 2, not type 1, it is type 2. Then the first animal produced through cloning was sheep. Answer is option 3. Thermal cycler is used for. Thermal cycler is PCR machine. It is for amplification of DNA. VNTR is variable number of tandem repeats. Answer is option 2. Which of the following scientists were awarded Nobel Prize in Chemistry in the year 2020 for the discovery of CRISPR-Cas gene editing technology? So it was Charpentier and Doudna. Answer is option 1. Then the first crop plant whose genome was completely sequenced was Oryza sativa. Then Sanger's method of sequencing DNA is known as chain termination method. The cell fractions obtained by differential centrifugations are in the following order. So they are nuclei, mitochondria, microsomes, ribosomes. Answer is option 2. EDTA is used as chelator of the following ions. It is Ca2+ and Mg2+. A typical polymerase chain reaction requires DNA polymerase, DNTPs, primers and template. Answer is option 3. Which of the following file formats can be used to store protein sequences? That is FASTA. Which of the following is a 6-base cutter restriction endonuclease? BAMH1. Yes, of course it is a 6-base cutter. Which of the following techniques does not require any kind of gel electrophoresis? Western analysis, yes, SDS page is needed. Northern analysis, agarose gel. Southern analysis, also agarose gel. Microarray analysis doesn't require any kind of gel. So answer is option 4. So thank you for watching my channel. Please do like, share and subscribe. Thank you.